excited to have Dr. George Waldbusser, Assistant Professor at the College of Earth, Ocean, and Atmospheric Sciences of Oregon State University with us. Before moving to OSU in 2009, George was resident at Chesapeake Biological Lab in Southern Maryland and obtained his PhD in 2008 examining the effects of benthic fauna on pore water advection and biogeochemistry in locations from the Pacific Northwest to the southeastern U.S. He conducted his first oyster research examining recruitment dynamics in the Hudson Raritan Estuary in New York City in the mid-1990s. George's long interest in human-environment interactions and the interface between ecology and biogeochemistry led him to begin working on ocean acidification impacts on marine bivalves in 2007 with Dr. Mark Green. George serves on the editorial board for the Journal of Shellfish Research and as an associate editor for Limnology and Oceanography Methods. Outside of his academic career, George grows organic apples and has been experimenting with different hard cider recipes for the past 5 years. George, I'm going to hand the control over to you for the presentation.
Great, thanks, Paula. And just as we get started here, want to thank the National Science Foundation for funding this work and the NOAA OA program for hosting this seminar webinar series. So before I get started, I need to acknowledge a huge number of people who've been involved with this work that I'm going to present here, particularly Burke Hales, Chris Langdon, who have been sort of critical in both the sort of chemistry and the larval rearing aspects of this work, and then a suite of postdocs and graduate and undergraduate students who have been really crucial in actually carrying out the work. So, so what I'm going to talk about today is really how the work we've been doing to understand larval bivalve responses to ocean acidification, and we're focusing on larvae because we know that they are probably the most sensitive stage for many species and really a population bottleneck, and we'll talk a bit more about that. So, and then just down in the center here, this is one of my favorite scanning electron micrographs that we've collected so far. This is actually an Olympia oyster larvae that is developing a shell, and it just looks similarly like Pac-Man, so I couldn't help myself to make a Pac-Man reference here. So we have the Olympia oyster here developing a shell, and a fully shelled Pacific oyster here. These are not to scale, I should say. So I just want to give a quick outline of the talk, and so first to start with some background and context about ocean acidification, variability, and environmental change, so really thinking about coastal and estuarine habitats. Then moving into why saturation state could and should matter to bivalve larvae, and then talking about the experiments we've been doing where we decoupled carbonate system parameters to understand what in fact the bivalve larvae are sensitive to. And then add on to that a little bit and talk a little bit about nativity of mittelid larvae. And so we've looked at native and non-native larvae to the Pacific Northwest here and examined their responses. And then sort of wrap it up with thinking about OA as a multiple stressor in itself. And then sort of bring it home with the mighty Olympia oyster, so which I'm pretty excited about some new data we've collected there. Okay, so Wei-Zhong Kai gave a great webinar a couple weeks ago, and so I'm not going to revisit that really too much, but I think just a couple important points is that really acidification in the coastal zone is we can think about that as sort of the weather that the organisms are exposed to, the same way we think about climate change and global warming and the effects of climate change on localized weather. We know that there are a number of processes that are going to affect carbonate chemistry in the coastal zone, and these processes are generally additive, and so even though chemistry can be, or CO2 levels can be higher, we know in the global ocean areas where they have already high CO2, their increase in CO2 is increasing at the same rate as places that have lower CO2, and so that baseline shift it appears to be ubiquitous across different variable environments. So just because the coastal zones are more variable, it doesn't mean that the baseline shift doesn't matter. And I think a great example of this is some work that Brian Helmuth has done looking at the temperature exposure of mussels in the intertidal zone. And so what I'm showing you here is sort of average temperature, and these are for data loggers that were put out inside mimic mussel shells across nearly 10 years in the intertidal in Northern California. And what I want to highlight is 2 years in particular here. And you can see the running average means for 2004 and 2007, roughly appear to be the same in what they're experiencing. So the mean conditions are pretty similar, but what becomes really important is if we look at extreme events. And so these are now on the same x-axis with year on here, and the number of days above 30 degrees C, which is a temperature that starts to create real thermal stress for these organisms. And what you'll notice in 2007, there were nearly 27 days above 30 degrees C where there was some thermal stress versus 2004, there was only about 9 or 10 days. And these numbers associated with the dots are just the frequency of return events. And so I think this really, the work in the thermal temperature groups that have been looking at physiological responses, some of them have really sort of, I think, provide us a path to be thinking about this a little bit more coherently, and that while climate change is a global phenomenon, to an organism, all relevant environmental changes are very local as the organism moves through space and time. So this brings us to some conceptualization that I've done with a paper I wrote with Joe Salisbury, and thinking about what contributes to that weather patterns that organisms experience and the perturbations of those weather patterns, so to speak. And so this is essentially just a cartoon, a hypothesis, if you will, of the different wavelength of processes that affect carbonate chemistry in any given area. And so starting from kind of tides at about the 12-hour time frame up to tens of thousands of years in geologic, to hundreds of thousands of years in geologic time. And I think the key thing to think about is that on these same process time scales, there's biological organizational levels from shell formation to larval periods in the orders of days to weeks out to sort of evolution adaptation occurring at these sort of longer time scales. And so what we know is that the frequency and magnitude of the contribution of these changes are smaller we get out to these longer time scales in terms of what the organism experiences. But the permanence of the perturbation also increases as you head out to those longer time scales. So we know when there's been big geologic change in the CO2 of the planet, there's been big changes in the composition of species that are present. So what I really want to focus on now is thinking about the bivalve life history and bottlenecks in that are really, we know, are related to sensitive areas in the life history for ocean acidification or other environmental stressors. And I just want to highlight 3, and then we're going to focus on really 1. And so the first is really this development of the dehinge shell, which is really what we've been focusing a lot of our work on. Previously, I've worked a lot of the work I did with Mark Green. I was looking at this period from the swimming to the settlement stage, and again, another bottleneck has to do with big developmental changes in energetic loads. And then finally, habitat, which has been the focus of a lot of restoration work and sort of persistence of shells, and we have some new work that's been funded to start looking at that as well. But what we're going to talk mostly about today is this bottleneck occurring in the development of that dehinge shell, because we know that this is a very sensitive stage for most invertebrates, and bivalves in particular, and it is a bottleneck for populations. So really the question we're trying to get at is what component of the carbonate chemistry matters most in bivalve larvae, and why does it matter? And so one of the big differences between today and the past in terms of CO2 changes is the rate at which it's occurring. This is some really nice modeling work that's been done by Barbel Hanisch and others. And what this is showing is essentially different simulations of a doubling of CO2 in the atmosphere, going from about 10 years to 10,000 years. And what you'll see is that the faster that increase in CO2, the faster or the more the mean saturation state of the ocean drops in the surface water. And so if we increase CO2 much more slowly, which has generally happened in the geologic time, the saturation state more or less stabilizes. We do get a decrease in pH, which isn't as bad. But that's really the big difference about what's happening today and what's our previous geologic periods. It matters also because in coastal zones and estuaries, freshwater inputs will change alkalinity. And we also know that many estuaries have different alkalinity end members and rivers coming in, so that becomes important as well to understand what components of the carbonate chemistry. Lower alkalinity waters will basically respond more readily and severely to increases in CO2. And then finally, the variables in the carbonate system don't respond linearly to increasing CO2, and so this was some work from our recent paper in Nature Climate Change, just illustrating what happens to pH and saturation state as CO2 and PCO2 increases. And what you'll notice is that there's a more rapid decrease in saturation state initially relative to pH. And we just annotated on here some aspects from the literature where we start to see chronic effects on bivalve larvae and where we start to see more acute effects at different saturation states. And this delta 0.3 pH unit is a value that's been banded around in some of the meta-analysis that have been conducted looking at responses. And so what really is that, and what's important is that almost all of the previous work that's been done on this has really bubbled CO2, and all the parameters change in some way, and there's very little ability to differentiate what really matters to the bivalve larvae. So this should start to lead us to understanding what traits ultimately result in resiliency or susceptibility of bivalve larvae to acidification, and we'll come back to revisit this. But there's a number of things that have been talked about in the literature so far. One of the big ones is the ability to compensate internal acid-base chemistry, and this is a primary mechanism that's been shown very clearly for adult organisms. Energy availability, and so having access to more energy is certainly really important. That's going to help to some point, and there's physiological limitations to that. There's a great example that gets thrown around about clam or mussels that live at hydrothermal vents in these highly corrosive environments. And it turns out they're chemosynthetic, and so they essentially have an unlimited supply of energy from the sulfide leaking out of those hydrothermal vents. And so that's probably one of the mechanisms that allows them to live in a really highly corrosive environment. Shell mineralogy has been talked about quite a bit, and that's really the thermodynamics, and I think that in most cases we've seen very little evidence of that in experimental data, as that being a determinant of susceptibility to ocean acidification. And then a lot of the work our group's been doing recently is to understand the role of kinetics, or how rapidly shell formation is occurring, and particularly focusing in on this early larval stage because it is a very rapid shell forming period. So really, I just want to everyone here and say, let's not forget natural selection works on traits, and thus it should be a key to understanding what OA ultimately holds for marine species. And so we need to be thinking about traits and what's really going to matter to the organism. So here's a suite of possible traits we talked about, or I mentioned, and we're going to just really kind of get into the kinetic side a bit more. So first I want to start with why saturation state should or could matter to bivalve larvae. So this is just the standard idealized model of organismal physiology under OA. It's really nice work that's been done by Hans Portner and many of the other physiologists working on these sorts of questions. And the sort of important features of this kind of model is that the calcification surfaces are within the body, so they're protected from the outside environment. Everything is linked through metabolism, and really it's the regulation of the internal acid-base chemistry that is key. So according to this model, only the external factors that affect the internal acid-base chemistry can affect the organism. All responses are generally thought to be related to how quickly bicarbonate accumulation occurs, and so how quickly can they compensate that the organisms can compensate that internal acid-base chemistry. And all the work that's been done so far shows that this generally occurs over the course of a few days in kind of the fastest responses that have been noted. So at least takes them a few days to compensate for this change, and we know from plenty of measurements in coastal environments that within a day the changes can be quite significant because of diurnal variability, production, and respiration cycle. So this clearly is an important mechanism, but it may not be capturing everything. I just wanted to highlight, this is a new study just came out last week out of Donald Monahan's group by Penn et al. This is in Proceedings of National Academies, and really the key thing that they found here, and these are sea urchin larvae, was that these are looking at essentially the energy budgets, or the ATP allocation to different components of the energy budget. The black areas are in these pie charts are to protein synthesis, and the gray is to proton pumping, so sodium, potassium linked ATPase activity, and the white is sort of everything else. And basically what the argument is, and it's really nice work, shows that under acidification stress in these red circles here, there's a really significant increase in the energy allocated for protein synthesis. And so clearly proton pumping and acid-base regulation is being upregulated, but there's a significant portion of that energy that's being reallocated to just simply synthesizing and regenerating protein in some way. So it's clear, here's one example, it's clear it's not just intercellular pH regulation, and I'm not saying that's not important, but we have an evolving model of understanding this, and I think in order to understand these responses, we have to be open-minded that there are more than one potential avenue for acidification effects on these organisms. So the work we've been specifically doing is looking at early shell formation, so we call it the Prodiscoconch 1 shell, and some people refer to that as dehinge shell. And just to summarize, and I'll show you some data here in a minute, is the reason this formation, this stage is so sensitive to saturation state in particular is the calcification surfaces are exposed, and so I'll show you those isotope data in a second here. We know there's limited energy until they develop that shell and they can attach the velum and start swimming and feeding. And the calcification during this period is extremely rapid. And so this is some isotope work, stable isotope work that's been done by a former graduate student of mine, Elizabeth Brunner. And what you're looking at here is sort of, we've sampled, these are Pacific oyster larvae sampled over development from fertilization, a couple days or a day after fertilization, out to roughly settlement or competent to settle. And sort of the key thing in the top graph here is to note that in the shell there's a much higher, heavier del-13 signal, which indicates greater incorporation of DIC from seawater. If this was 100% DIC that they're putting into the shell in aragonite, it should be about, I think about 2.7. And so what we can do is, because we know what the metabolic signal should be from the food, we can calculate the proportion of metabolic carbon in the shell. And so what we notice is that 2 days, there's about 7% metabolic carbon in the shell, and by the time that they're ready to settle, that's up just over 14% metabolic carbon in the shell. And so we interpret that in part as being these early surfaces are more dependent on DIC. These early shell development is more dependent on DIC and it's more exposed to external seawater. And as they grow and develop new organs and the ability to close off those calcification surfaces more readily, then you get more of the metabolic carbon in the shell. It's also important to note that at this stage, at this early stage when they're building the shell, their mass-specific metabolic rate is almost an order of magnitude higher than what happens later. So they're respiring at a phenomenal rate, but most of that is not being captured in the shell because we believe we think this data shows us that those surfaces are more exposed. The bottom graph, just to point this out, all this really shows is that from the early period on, they're more dependent on the maternal carbon derived from the egg, and as they start feeding, that proportion goes down, and there's still measurable amounts of maternal carbon in those organisms out to just about a week and a half. Okay, and just finally to show this rapid calcification on the right here, this is about a 6-hour time frame. They go from an unshelled larvae, you can see the back of the hinge here, and this is the initial periostracum that's formed, the outer organic coating of the shell, and within about 6 hours that has been fully formed and calcified. So what we're arguing is that saturation state sensitivity is really about the kinetics. It's because this rate of shell formation is very fast. So this is some work, again, by my former grad student, Elizabeth Brunner, and this is just showing our estimated calcification rates, which are very conservative, actually, relative to the more recent SEM work we've been doing. But if we take the averages over days, we can see there's about a really big drop in calcification rate from that early calcification of that first shell onward. And what that implies is that if we take that calcification rate, and this is the abiotic equation for calcium carbonate precipitation, we take those calcification rates and solve for the rate constant needed under a decreasing saturation state, what we see is that that rate constant increases exponentially, and it increases much more rapidly at higher rates. And so what we're clearly saying is that this isn't the equation that defines biocalcification, but it's a constraint on the system that biology clearly overcomes, but it's a physiochemical constraint on the biology that they have to contend with in some way. So I guess the analogy I've thought of recently is that birds fly in spite of gravity, but that doesn't mean that it doesn't exist for them, right? So those physical forces are still there, the physical chemical forces are still there. Biology is very good at overcoming them, but it's still, there's a cost for doing this, and that's why it becomes we believe, much more energetically expensive to calcify during this early period when those calcification rates are high. And these rates are hundreds to 1,000 times greater than an abiotic precipitation rate. So there's clearly an acceleration that goes on during this period and throughout the later life stages of bivalves and all other calcifiers. So the egg reserves then provide that initial energy for the shell formation, and so really the egg size, that's the larval lunch bag. I'm just going to move through this to keep things going, but we see egg lipids are high. They drop during that initial period. They start feeding and then accumulate those again, preparing for settlement. So its calcification is clearly biological mediated and requires energy. There's proton pumping to maintain these the saturation state in these calcification surfaces, whether that's pumping protons out of hemocytes or out of an internal surface area or a space where they're calcifying from. And then there's also the organic matrix, which is the sort of scaffolding or the framework for the shell that they must produce, which is actually the bigger of the two costs, which is exciting in the context of the findings of Pan et al. Showing that there's a big increase in protein synthesis under OA stress. So I wanted to show you a little evidence we have for less effort in the initial shell. We tend to think about it in my group as the construction trailer when they make that initial shell. And once they get that set, then they can start building a better shell. And so here's a cross-section of a larval shell from my group, and you'll see this initial shell layer is up here in the top. We've done some transmission electron microscopy on this, and as the beam resides across this entire section, what you'll see is that this calcium carbonate is softer in this initial shell, so this is the same exposure to electrons here, but we see in that initial layer that's deposited that this beam damage is much greater, and we see that consistently across the board. We've also done some dissolution experiments on larval shells, so these are not live larvae we're putting into acid. We've cleaned the larvae out of here, and we're just looking at the sort of pitting and where the shell dissolves. And basically what we see is that the mineral acid preferentially pits and dissolves that initial shell area, the Protisconch 1, and it appears to be more dissolvable than other larval shell microstructures. So we're going to really focus on the formation of this Protisconch 1 shell, which I've highlighted here in this electron micrograph in these Pacific oyster larvae. So this is the shell that gets put down in that first really rapid period for the Pacific oysters. And then show you some data from experiments we've been doing to illustrate the saturation state importance of this at this stage. So what I'm going to start showing you now is experimental data we've collected with our group, the work we've done at the Hatfield Marine Science Center, where we've decoupled the carbonate parameters to understand larval bivalve responses, particularly at this sort of first stage, going from an unshelled developing embryo to a shelled swimming straight-hinged veliger. So this is just a couple examples of some of the experiments. I'm going to show you data from Mytilus galliprudentialis, is the Mediterranean mussel, which is an incredibly important aquaculture species that's grown here and elsewhere. And then Chrysostrea gigas, again, the Pacific oyster, and is also non-native to the U.S. Northwest coast, but also sort of the key oyster for the oyster industry on the West Coast here. So what we've basically done is manipulate the DIC and alkalinity to decouple the carbonate system parameters. And so what I have plotted here in the y-axis is the PCO2, and on the x-axis is the saturation state with respect to aragonite. The gray circles and squares are different experiments, and what these show are essentially our treatment values. So we've done this 4 levels of CO2, 4 levels of saturation state with respect to aragonite in a factorial design. And what you'll notice is that on these isopleths or pH, and so what you can see is we can't decouple completely the 3 without a third manipulating factor there, which becomes exorbitantly big experiments to run. These are already large, very complicated experiments to put together. But what you'll notice is that we can generate conditions where we have low CO2 and low saturation state, high CO2, high saturation state, and all sorts of combinations in between. And then we also have conditions where both the CO2 and saturation state change, but we have constant pH on these isoplats in many cases. So it allows us to essentially evaluate the role of these three different variables on the effects of the shell development and some other physiological responses. So just to give you a quick background on the methods, these are all closed bottle incubations. For everything other than the last set of experiments I'll show you, they're 2-day incubations for the mussels, and for the Pacific oysters we do about 5 days. For the Olympia oysters, that take longer to develop in that same period. We have 16 chemistry treatments. We have 3 replicate BOD bottles per treatment. We do 3 counts per each bottle. And then what we're doing primarily is measuring shell length. We do triplicate counts for development. And so we're scoring whether the shell is developed normally or not. And we're measuring shell length of all the normal larvae that we collect in those counts. And so we end up scoring about 100 larvae per subreplicate, which means about 300 larvae per BOD bottle. And so and we have about 60 BOD bottles, including controls. And so we have a really very large sample size to sort of put together some of this data. And I think the important thing I really want to point out here is that we're only measuring acute effects. And so we're only measuring how this affects the ability for the larvae to develop and grow in that first kind of developmental period. So I'm going to show you a series of plots that look very similar to this. On this set here, again, we're going to focus on the Mediterranean mussel and the Pacific oyster. I have on the y-axis for all of these plots the proportion normal. And on the x-axis on the top set is PCO2, the middle scale is pH, and the bottom one is saturation state. These data are published in our Nature Climate Change paper, which came out in print in the March edition. And it's basically proportion normal, so it's the number of larvae that build a normal shell at 2 days relative to the total number of larvae that we count in that sample. And so what we find is that at extremely high CO2 levels, we see normal shell development on par with what we see at lower levels, up to over 3,000 microatmospheres, almost 3,500 microatmospheres in some cases. We get relatively good shell development. pH, I should point out that these shading are essentially the saturation state treatments, so darker symbols indicate lower saturation states, and so you can see where we're going with this. But for both the mussels and the Pacific oysters, we see in general there's very good development, even down to a pH of 7.4, saturation state still high. The mussels appear to be maybe a little bit more sensitive to pH, but again, a lot of this has to do with the covariance with the saturation state. And here the picture becomes very clear in the response to saturation state. We see very clear threshold response. It's repeatable. So each of these, we've run these experiments twice for both species, and we see very, very similar responses. So really for this early stage, this early shell development, saturation state is a primary variable of importance for these larvae. And there's really little to no impact that we can determine from pH until you get to these really low pH values, and typically only when conditions are undersaturated with respect to the mineral. So I'm going to show you data on shell length, and so these are basically measuring how big the shells of the normal larvae are. So we're excluding anything that's abnormal because we're trying to not bias the results and look at sort of, okay, if they can develop normally, does it then not matter at all? And so just to pull these up, we did these only on one of the 2 sets of experiments. And what you'll notice again is we see at really high CO2, really no effect on shell length of the normal larvae, same thing with pH, but it becomes very clear in this saturation state space that that's what's actually driving the response of shell length. And so the decrease in saturation states makes smaller normal larvae, and what this really indicates is that a smaller scope for growth, meaning the shell is more expensive to make, or they're using the energy somewhere else during this initial shell building period. You might look at this and notice these little oddities. So for the high saturation state, it actually looks like pH is having a negative effect, or the CO2 seems to maybe have a slight positive effect. Those are probably real, and that's probably related to total DIC concentrations. And so that's something that's been shown to be important in coral calcification, and so increasing the amount of DIC seems to help corals grow more quickly, and it might be true here as well. But again, that is a really small effect relative to the saturation state effect that we documented. Okay, so we're going to move on and compare mussels from Portland or from Oregon, so our mussels here with really nice, well-formed, groomed beards, and our Gallo provincialis mussels, which were actually collected or gathered from Carlsbad Aqua Farms down in Carlsbad in Southern California. So they have very different exposure histories. You know, the chemistry down in this region of the West Coast is much more stable. It's not subjected to the strong upwelling pressures we see in the North Coast. And so if we anticipate that exposure to these different environments might pre-adapt these organisms to some kind of sensitivity or make them more stronger, then we would expect the Californianus mussels that are found in Oregon to be more resilient to ocean acidification. And in fact, it's not what we see at all. So these are the data from the previous experiment I showed you for proportion normal and also shell length for the Gallo provincialis in white and gray and the Californianus in black. And you'll see there's really no discernible difference between their responses in proportion normal development. And we see essentially this very similar response between them with perhaps slightly higher growth rates at higher saturation states for the Gallo provincialis. So we're taking this to indicate is that hanging out in an OA hotspot such as the Oregon coast where we have really strong upwelling doesn't seem to help Californianus, at least in terms of this early developmental stage. Okay, so a couple more slides here. OA is a multiple stressor, so there's been a lot of interest in thinking about multiple stressors in the environment. And so what we've done is take those same experiments where we've looked at shell development and shell length, and then also measured respiration rates and feeding metric for Californianus, or the native millipede here on the Oregon coast. So similar plots again. This is respiration rate on the y-axis now, PCO2, pH, and saturation state. We had to do this on a reduced set of treatments because it just became logistically impossible to do this across the entire suite. So what we focused on was trying to get a few values in the mid-pH range that have different saturation states, and then a high pH and a low pH. And what we basically see is that pH is driving the show, and this is basically what we'd expect. And so we can say, and this is clear from other work as well, that pH is actually affecting the respiration rate. It's not saturation state and it's not CO2. So even at high CO2 rates, if pH is low, as here, there's really no discernible effect of CO2, and if saturation state even well undersaturated, we see no effect on respiration rate. It's only when we have that really low pH. We don't have any measurements between 7.8 and 7.4. So we don't know if this increases linearly. We don't know if it makes a step function here, but we do know that the CO2 or the pH is affecting respiration rate. And then this is a feeding metric we'll call proportion feeding. So this is at 44 hours post-fertilization on larvae that have been exposed to the same condition treatments, and we're measuring with some techniques that Matthew Gray and Chris Lang have been working out with using beads to look at the proportion feeding. And so the strongest effect here is really due to CO2. We see potentially some slight effects of pH at the really high CO2, but generally speaking, it's really appears to be a CO2 effect on the number of organisms, number of larvae that are feeding at this early stage. So I just said all that. So then I'm just going to wrap it up here with the mighty Olympia oyster. And so these are basically the same experiments as above, and so I'm going to show you shell development and shell growth, but we've extracted the brooding larvae to measure their responses under the same developmental phase as the other organisms. And so the difference is Olympia oysters brood the larvae for 1 to 2 weeks before they're released, and so they maintain within the maternal brood chamber. And so there's been very little work done on that really early stage, and so some really great work that Matt Gray has done down at Hatfield worked out the techniques to be able to extract day-old fertilized Olympia oyster embryos and develop them, grow them normally, and we find basically no effect of their on development and growth if they're outside the brood chamber than when they're inside under kind of normal conditions. And so and I'll show you some controls for that as well in these data. So basically what we see for the Olympia oysters is really almost no response to any of the variables. Again, these are the same graphs, CO2, pH, and saturation state, proportion normal here on this group on the left, and shell length on the right. We see what might be look a little bit like a negative response as we get to these really high saturation states, and we haven't really figured out what that is. But we also ran control, so we have controls for all of our experiments where we keep them under kind of good water, and we see that even with the brooded larvae that we extract later at the end of the experiment, that there's really almost no difference between them. So this doesn't really mean that Lurida are impervious to OA, it's just this early developmental stage. And again, I'd point you to the really nice work that's been done by Annalise Hediger et al. When she was down at UC Davis on carryover effects in Lurida from exposure. And so this really got us thinking about what is it about Lurida that gives them some trait or some resiliency to OA. So I'm going to show you this, and I showed you the SEMs previously, but these are light microscope images and cross-polarized light microscope images at 18 hours, 24 hours. So this is a different cohort than I showed you previously, but it shows about the same 6-hour window from where we first start to see some initial calcification, which shows up as this glowing color, all the way out to a fully calcified shell. So this happens in about 6 hours. For Lurida, this takes about 24 hours. And so these are, again, light micrographs, cross-polarized light showing where the calcium carbonate is forming, and we know it forms, starts at the hinge of the shell and extends outward, and they get to be about fully calcified in 24 hours. There are the concurrent SEMs, and for this group in the same time frame, you can see that sort of puffing up where they're starting to fill that shell out, and then within about 24 hours from when that calcification is initiated, it actually completes and they form that shell. So we've actually measured the calcification rates on the larvae, so we've counted out the larvae and measured very carefully the total calcium content. And so these are the Pacific oysters during this early shell development period, and the Olympia oysters here. And what we basically see is that the Lurida calcification rate is about 7 times slower during the same developmental stage. And so although the timing is different, this is when that same PD1 shell formation period begins. I'm not sure, these are a couple of measurements we had here. There's a lot of error in these measurements, and I'm not sure how to interpret them quite yet, but I've left them out of this regression analysis just for the sake of this presentation here. So what we've learned so far is that saturation state does matter to bivalve larvae, and if embryos can't develop, then there are population impacts, clearly. If the larvae can't get past that early stage, nothing else really matters later on. OA appears to potentially have multi-stressor type effects across bivalve life history, but again, that saturation state matters most first for that developmental period. But I think this is really a key issue when we think about coastal zone acidification, because all these estuaries are going to respond differently with these different drivers. And so although I'm not advocating against the other multi-stressors, thinking about oxygen and temperature alongside and salinity, I think what we're sort of pointing towards is that we really just starting to truly understand the responses of some of these organisms in these environments. Nativity to an OA hotspot in itself does not convey resiliency, and we saw that with the middellids. However, slow shell development during PD1 appears to convey some resiliency and supports our idea of a kinetic hypothesis for this early larval sensitivity. And again, traits are what natural selection acts on, and so the rate of shell formation during PD1 might be a critical one at this stage, is what our argument is. So what we're doing now to follow up some of this work is we've now set up and beginning trials on a flow-through system that we can explore these same types of questions where we decouple the carbonate chemistry, and we can look at how pH, saturation state, and CO2 affect the organism over the entire larval life history and track them out all the way until settlement. So I've run a little bit long, and so that's about all I got. And thanks for listening, and I guess I am now ready to take questions via the chat. So, okay, thanks.
Thank you very much, George. That was a wonderful presentation. So yeah, we're going to open the floor now for any questions for George, and if you could type your questions or comments into the questions box on the GoToWebinar control panel on the right of your screen, and we already have at least one question from Libby Jewett, and she wants to know, she just makes a statement. She says maybe Middleus californianus just hasn't adjusted to the new ocean yet, or adapted to the new ocean. If you reduce the habitat such that newly set spat are in perhaps acidic mud instead of up in the water column, I presume that would also cost the spat energy for shell formation. How are you able to parse out differences in feeding growth, etc.? I'm sorry, that was another question.
Okay. Well, let me start with the first one from Libby. Right. I guess we've always had an upwelling system here, so they've always been exposed to elevated CO2 to some level. And the gallows are originally from the Mediterranean, which has very stable CO2 and much higher alkalinity. The Mediterranean mussels from these experiments from Carlsbad have been there for 20 years, and they've been confirmed with genetic testing that there's no hybridization. So they've been exposed to very different conditions. Clearly the conditions are getting worse, but if they've already been exposed and are exposed to upwelling conditions well before we're putting CO2 into the atmosphere. So I guess we would anticipate that in that environment that they would have adapted in some way to that higher CO2 that's there. And I think, so there, and then there was a clam question after that.
Evan asked, how are you able to parse out differences in feeding, growth, etc., due to pH versus PCO2?
That's the whole. That's the whole point of the experiments. And so let me try to go back here. If you're whoever asked that's on the, if that's Libby. But basically we essentially we do this by we're manipulating the total DIC and alkalinity in the experiments. And so by changing the total amount of those and the ratio of those, we can create these different conditions. And so we have conditions where we have. You can look across, say, on this diagonal here of 7.7 on the left graph that I'm pointing to now. We have a constant pH, but we have a CO2 that increases from 500 to well over 3,000. And we can do that on any one of these isopleths for pH here. And so it's the experimental manipulations, and it's the full factorial design with 4 levels of each of the CO2 and aragonite saturation state that allows us to do that.
Okay, thank you. Here's another question from Libby. Have you tried experiments crossing OA and hypoxia yet?
No. Nope.
Okay.
Yeah, we haven't, and just because that would put another, like I said, we have usually somewhere between 60 and 70 BOD bottles, and to do these counts, each experiment we end up counting somewhere around 6,000 to 10,000 larvae per experiment, and so if we add another factor in there, but certainly you'd expect some other effects there, and so we could, in theory, you could do this and put hypoxia on top of saturation state and pH and CO2 and see what those effects are. But it just starts to increase exponentially the number of containers and experimental size that we're doing, and we already have a fairly large team. But it's a great question because it'd be really neat to look at whether or not the hypoxia effects are different when you tease apart these different carbonate system parameters.
Mm-hmm. Okay, Jim Pilardi asked, you've shown data on shell length normalcy, but do you have any data on larval survival across the various carbonate system parameters?
We take the development as the survival. Usually once they are malformed, they usually don't survive. It's a toxicological assay that's been used for decades, and that's part of the point of the new experiments. It's a great question, and it's part of the point of our experiments we're spinning up now for the longer term studies to see how many actually make it and are able to metamorphose and settle. And so I guess I'd anticipate that even those smaller ones, even if they're normal, that they'll make it, they might very well could make it to settlement, but often what the hatcheries have seen is they will often see, and we know from natural populations that there's often a lot of mortality that occurs during metamorphosis because it's a highly energetic period, and if their energy stores are already hit because they've been subjected to some of the stress, then we would anticipate that. And I believe some of the work by Chris Gobler has shown that as well, that you can see fairly good survival through the larval period. that metamorphosis tends to knock them out. And I guess it's what I'm really curious about is whether or not that effect on the saturation state of the early development, how much that becomes important later on. So we would anticipate a sort of sublethal effect of pH during that larval period, and maybe the saturation state becomes a little less important. And what we're hoping to do with these longer-term experiments is really tease those apart.
Mm-hmm. Okay. Robert Brock wants to know what actions, if any, can be taken as a natural resource manager to reduce impacts of ocean acidification on shell-bearing organisms.
Well, I guess the first one is we reduce carbon emissions, but I think we all know that is a challenging prospect in itself. There's a lot of great work going on looking at mitigating effects, and so the work that's been done in hatcheries at least with buffering, but clearly we can't buffer the entire ocean, or it'd be really difficult to do that. I think that there's some work that's just gotten funded up in Washington State looking at growing macroalgae in association with some of the culture work, oyster culture in the environment. I mean, I guess overall I would say that, and we're doing some work with shells now to try to understand the role of sort of shelly habitats. Those are often removed and are highly diminished from what they once were, and they provide a potential buffering source in shelly environments. But I guess ultimately, we've done work thinking about policy issues and dealing with sort of the other drivers of coastal carbonate chemistry, and so reducing eutrophication. But really getting at restoring the resiliency in coastal ecosystems and taking a whole wide holistic habitat of restoration and restoring habitats, I think, is really a crucial one.
Thank you. Matt says, thanks, George, for your talk. And Dick Feely says, excellent talk. What do you think might be the impacts of natural populations of oysters and mussels over the next 50 years? Are these different species of larvae in the water column? Oh, that's another question, sorry.
Yeah, it's a great question, and I think we're just starting to get some ideas about how to get at that. But there's interesting differences in the reproductive biology between the primarily aquaculture species, the Pacific oyster and the native oyster here, and so I think it's going to take time to discern those differences. And there hasn't been a lot of focus on the post-larval stages. There's been some work clearly that's been done, and we're spinning up some work now as well to try to look at those exposure histories. So, I would never say that all the oysters are going to die. But clearly there's been recruitment issues in Willapa Bay, and I've been involved with some work that Burke Hales has in review now. And this is just one of many factors in all these coastal environments that are going to impact the organisms. And so I would say the responses that I'm showing here, we've removed all the other potential effects that are going to occur in those environments, including hypoxia, different thermal regimes, changes in food availability. So these are kind of the best case scenarios, I think, of what organisms how they will respond to OA. The other thing to point out too is that in all of our experiments, even at some very low conditions, we do get some larvae that develop normally. Now those are generally much smaller than the other ones, but there's potential there for breeding as well, and some nice work that Chris Langdon is doing, as well as Dennis Hedgecock, in looking at sort of genetics and breeding is one way. But again, a lot of this is manipulated, and we're talking about a very small number of species that have a high economic value. And there's lots of species out there that we just simply don't either know what their responses are, or there isn't the interest in them. And so I think it's fair warning that, we're going to see some impacts, and there are likely some things that might do better than others, but there's going to definitely be some losses of species if we don't address the emission issues.
Mm-hmm, mm-hmm. And Beth Turner wants to know, are these different species in the water column? Let's see, I'm sorry. Are these different species in the water column at the same time? Might there be impacts on differential survival or growth that would lead to one species having an advantage over another?
Yeah, that's a great question. I mean, there's a real, for the oyster species that we've looked at so far, the Pacifics and the Olympia oysters, they have very different breeding strategies, and they're very different cues for what triggers reproduction. We know much less about the Austria Olympia oysters. Chrysosteria need a certain temperature to spawn, and again, this work that's in review that Burkhouse has led, looking at Wilpa Bay chemistry and temperature data, seems to indicate that there's shrinking windows of time when the good chemistry and the good thermal regime overlap. Now, the difference with Austria is that they tend to reproduce much earlier. They don't need the warm water, and they tend to have peaks that sort of bracket to some degree the upwelling window. They will reproduce throughout the summer period of upwelling, but if they're reproducing before upwelling really kicks in and they get the larvae through those early stages, then they clearly have an advantage in terms of getting past those susceptible life history stages. But again, the challenge is we don't really have good time series data for a lot of these places to try to put together the reproductive periods, the larval periods with when the chemistry and temperature really look good for these things. So it's a great question. I think it's the next step for a lot of this work is to really understand that relationship between the variability, the seasonal variability in the chemistry, and the reproductive timing for these different organisms. Because if they do time it differently, then they might find a way to get past those bad periods.
Mm-hmm. Thank you. Jan Newton asked, do you think outplanting Olympia oysters is a useful adaptation strategy, or is that premature?
Yeah, I mean, there's great efforts going on in Oregon here already. There's in Etarts Bay, there's a native oyster restoration site, and there in Yaquina Bay as well, some work with Oregon Oyster, and the Nature Conservancy has been involved with both of those efforts. They've been putting out Olympia oysters, and there's also been some work in Coos Bay planting Olympia oysters, and I think in many of those cases they're really for restoration. And I think again many of the estuaries in Oregon and in Washington were once full of these Olympia oysters, and so I see no reason to not make efforts to restore that. And once those populations are in place, it gives us a better opportunity for those organisms to rebound potentially. I don't think we should give up hope and say it's all going to be bad, so we should stop trying. I think that's the absolutely wrong approach to take. So I would strongly advocate for that, and there is a small market even for Olympia oysters. Both Taylor shellfish and Oregon oyster produce Olympia oysters for market. So I know Oregon oyster is heavily involved in some of those restoration efforts as well. So.
Okay. Great. And this is the last question. And it's asked by Matthew Leibolt. Given the importance of maternal carbon to the muscles or the maternal brooding for the oysters, the health of the parent seems to have a big influence. Will sublethal stresses to the parents necessarily affect the larvae, or are there other traits working to help sick parents have healthy larvae?
Yeah, that's another great question, and as we do more work, we can start to put some of the story together better. There's been some work out of the Australian group led by Laura Parker where they've looked at maternal effects, and so what they found with Sydney rock oysters was some I would say sort of weak effects anyway of exposure during the maternal phase seeming to convey a little bit of resiliency for the larvae. We've done some work here with Pacific oysters, which hasn't fallen in line with that. So even short exposure windows for the parents for Pacific oysters seem to result in poor development and growth for the larvae. And so it's certainly another aspect of this. And those exposures that we did with those experiments were only 2 weeks long, and so I think it's another component of this, and the hatcheries have known this for a long time, so they look at things like egg size and quality, but it's certainly another component of this problem. But in general, those adults are going to be more resilient, I would say, and I don't know that we have a good test yet of how important that is relative to the exposure, and I think until we start getting more integrative across the life history and doing longer experiments where we measure all of these things, it's hard to really put those things together and assign importance or value to how much each of those are relevant to the organism. That's a great question again. There's lots of questions to be asked. So.
Yeah, there are, and in fact there are a few that we're not going to be able to get to today, but what we'll do, George, is we will send those to you and then get you to answer those, and then we will post those online with the presentation in the PDF.
Yeah,
so again, thank you very much for a fantastic presentation.
No problem.
Thank you, yes. And again, if we didn't get to your questions, we will get those to George and have those posted. And if you are interested in joining the SOCAN listserv, you can find that here on this slide. And the webinar and the PDF will be posted at the first link that you see there. If you have any questions about SOCAN, you can contact the link there in the middle. And you can keep up with the latest happenings of SOCAN by joining the listserv, which is the last link that you see here. We'd like to thank you for attending the SOCAN State of the Science webinar today. It was most informative. We welcome any feedback or further questions or suggestions that you have for topics for this webinar series. You could submit input by replying to the follow-up email that you'll be sending in a couple of days, or by emailing us at one of these links that you see here. The recording of this webinar and a PDF, again, will be available on the SOCAN website that you can find listed on the press listed here. Our next webinar will be 2 weeks from today, on Tuesday, April the 21st. Chris Chambers of NOAA's Northeast Fisheries Science Center will discuss effects of elevated CO2 on early life stages of marine fishes and potential consequences of ocean acidification. That will begin at 12 noon Eastern Time. We hope that you'll join us. Thanks again, George, for this very informative presentation, and to all of you for your thoughtful questions and for taking the time to join us today. We hope to see you at SOCAN's next State of the Science conversation. This is the end of the webinar session.